Only 10 members.
Madam?
Only 10 members ఉన్నారు కదా సార్.
జాయిన్ అవుతారు, మధ్యలో వాళ్ళు జాయిన్ అవుతారు మేడం. ఎందుకంటే మనం ఇది వర్కింగ్ అవర్ కాబట్టి కొద్దిమంది వెనక ముందు జాయిన్ అవుతారు. మీరు స్టార్ట్ చేయండి మేడం. ఓకే, వెరీ గుడ్ మార్నింగ్ ఆల్ ద పార్టిసిపెంట్స్ అండ్ రెస్పెక్టెడ్ రిసోర్స్ పర్సన్ Dr.A.Shailaja మేడం గారు, అసిస్టెంట్ ప్రొఫెసర్ డిపార్ట్మెంట్ ఆఫ్ బాటనీ, గవర్నమెంట్ డిగ్రీ కాలేజ్, Siddipet autonomous, Siddipet district. Madam has used experience in the field of plant sciences, in the especially for all competitive exams. Last 2 years, Madam has given her valuable time for our CPGET aspirants, and they are get benefited. So, in this regard, we would like to invite today's resource person, Dr.A.Shailaja Madam Garu. Please welcome and share your screen and start your session, Madam. Please welcome.
Okay, sir. Thank you, sir. So today I am going to discuss about the anther structure and microsporogenesis in detail. So already we have completed that introduction of embryology and what are the parts in flower. Parts of flower completed. So calyx and corolla, androecium, gynoecium, the male sexual organs and female sexual organs. So today we are going to discuss about the case of anther. So see here. Sir, it is audible? The screen is okay? Sir. So the introduction about the anther. The stamen is the male reproductive organ of the flower. Typically, it consists of two distinct parts. So that is a lower sterile part, the filament, and upper petile part, that is called anther. So today we are going to discuss about the anther structure. Okay, case of anther. The sterile median part of the anther is known as connective. So that median part of the anther, that whereas where that is a lower sterile part, that is the filament, and upper part is called petile part, that is the anther. So in this flower, see here, so sterile part and petile part. So in the anther structure, that petile part is called anther, and the other lower sterile part is called filament. Okay. So now we are going to discuss about the anther. So in this anther, typically it is two-lobed, and that lobes are called anther lobes. Okay, so that two lobes are there. So in the two lobes, that 4 microsporangia. Each anther has the 4 microsporangia. Such anthers are described as dithecous. Dithecous means each anther lobe consists a pair of pollen sacs, that is, or pollen sacs or microsporangia. So that's why we are observed that in the anther it has 4 microsporangia. Okay, in some members of Malvaceae it has a bisporangiate, but bi means only 2 are there. Okay, so what is the development of anther? The young anther is almost oval in shape. It is composed of a homogeneous mass of meristematic cells surrounded by an epidermis. See here, this is the anther, and that outer layer that is surrounded by an epidermis. The outer line soon becomes more or less rectangular. That 4-lobed. In each lobe, few hypodermal cells become more prominent than the other cells. Their larger size and dense cytoplasm and prominent nuclei. Okay, so these cells from the archisporium Generally, the archisporium consists of vertical rows of cells. They appear like a plate of cells in the transverse section. Okay, so in the anther structure, so see here, this is a bilobed and dithecus. A longitudinal groove separate the theca. In the cross section, the anther is a tetragonal structure. It consists of 4 microsporangia, 2 in each lobe. Okay. So, latter 2 microsporangia of each lobe fuse as a pollen sac. So, see here, structure of anther. A microsporangium, that a mature anther wall is made up of following 4 layers, that is epidermis, endothelium, middle layers, and tapetum. Okay. So, in the anther structure, that in the anther wall, that is made up of following 4 layers. That layers are epidermis, endothelium, middle layers, and tapetum. Okay. That first is epidermis. Outermost layers that protect the pollen.
भड़का मत नहीं
Disturbances. Hello. Okay. Please mute yourself. Endothelium, middle layers, and tapetum. Outermost protect the pollen, help in descent of release pollen. Okay. Here, this is a connective epidermis, endothelium, sporogenous tissue. Okay. I think this is very clear to you. First one is the epidermis. This is the outermost layer of the anther wall. This is made up of tangentially and flattened. The epidermis perform its usual protective function. Okay, epidermis is for the protective purpose. See here, these are the pollen grains released from the anther. See here, first we will epidermis. It is the outermost layer of the anther wall. It is for protection. Okay. Next, coming to this is the this is the layer of the anther wall. Mostly, it is single layered, but sometimes it may be several layeral. Example: capsicum.
Madam, please unmute yourself. Madam, please unmute yourself. Madam, please unmute yourself. Madam, am I audible? Madam, please unmute yourself. Shailaja Madam, you are in mute. You are in mute. Please unmute yourself.
Oh.
Okay, please proceed, Madam. Proceed.
Okay, sir. Sir, hello. Sir. From the beginning, that is mute. Mute lo unda, sir. Only first one explain cheyana. Siddipet sir, please. Okay. So in the anther structure, that 4 layers are there: epidermis, endothelium, and middle layers and tapetum. So we are coming to the first epidermis. This is the outermost layer of the anther wall. It is for the protection. Okay. Next, coming to the endothelium. This is the subepidermal layer of the anther wall. Mostly it is single layer, but sometimes it may be several layers. That is example for that several layers. Capsicum and datura. Okay, capsicum and datura. So this one is the third one, the middle layers. Okay. Before that, the endothecium here, that endothecium, the cells of the endothecium are radially elongated and attain their maximum development at the time of anther descent. Okay. So that is the radial and inner longitudinal walls of these cells have characteristic fibrous thickening. So, and these thickening are U-shaped or ring-shaped. Endothecium, it formed as a U-shaped thickness. Okay. So that is the endothecium. It is the second layer of anther. And coming to the middle layers, inner to endothecium, 2 to 3 layers of pair, that is 2 to 3 layers of parenchyma cells are present. These are known as middle layers. Okay, these are known as middle layers. Next, coming to the, see here, these are the diagrams for this. Okay, middle layers, inner to endothecium. These parenchyma cells, 2 to 3 layers are present. These are known as middle layers, as they lie in between endothecium and tapetum. Tapetum is the innermost layer, and endothecium is the second layer. Epidermis is the outermost layer. Okay, so the middle layers may store food material in some taxa, which is utilized by pollen mother cells. That is middle layers. And the last one is the tapetum. So you see in this diagram, that is the tapetum. The tapetum is the innermost layer of the anther wall. It surrounds the sporogenous tissue and attains maximum development when the microspores are in tetrad stage, after which they collapse. So generally, already we are discussed in the introduction class about the tapetum. Generally, the tapetum is single layered, but it may divide periclinally and become biseriate, as in pyrostegia and trachoma. Okay, so this is the tapetum is a food supply. It is for food supply. Tapetum is usually a homogeneous layer surrounding the sporogenous tissue. So in some taxa, however, dimorphic tapetum is present. In this plant, the tapetum cells are present towards the protuberate side of the anther, or relatively small, and the tapetum cells towards the connective are large. So that larger cells are called a C tapetum, and some are the protuberate side of the anther. These are relatively very small, so we are called the P tapetum. Okay. Some are called a P tapetum and some are called a C tapetum. Because of that, the towards connective, these are very larger in size. And coming to the tapetum, types of tapetum. 2 types of tapetum is present in the angiosperm, amoeboid tapetum and secretory tapetum. This is also known as plasmodial tapetum. Amoeboid tapetum is also called as plasmodial tapetum. In this type, the inner and radial walls of the tapetal cells break down early due to the action of hydrolytic enzymes. See here. So, what is the change we are observed in the sporangium? That tapetum. The tapetum is 2 types. That is amoeboid tapetum and secretory tapetum. So, in the amoeboid tapetum, this is also called a plasmodial tapetum. In this, the inner and radial walls of the tapetal cells break down early, and due to the action of hydrolytic enzymes. Some of the performance of the hydrolytic enzymes, that cells are divided and break down. So that's why the protoplasm of these cells enter into the anther locules and fuses to form a common mass. That is called periplasmodium. Okay, that is called some mass of cells are formed. That is periplasmodium. The amoeboid tapetum is considered to be of primitive type and is found in monocots and in some lower dicots. So the example already I will discuss in the yesterday's class, that is introduction. Alisma, Bitomopsis and Typha. These 3 belongs to amoeboid tapetum. Okay. So these are the examples for the amoeboid tapetum. Alisma, Bitomopsis and Typha. Okay. That is the amoeboid tapetum. And coming to the second tapetum, that is secretory tapetum. See here. So some In this diagram, it's very clear. Endothelium and middle layers and microspore mother cells are formed like that. Tapetum is the inner side, innermost layer. The secretory tapetum, this is also known as glandular tapetum. This is also known as glandular tapetum. So amoeboid tapetum is called plasmodial tapetum. Secretory tapetum is called glandular tapetum. The cells of the secretory tapetum remain in their original position till the development of pollen grains. At the pre-mitotic stage of the pollen mother cells, tapetal cells are very thin and possess almost all cell organelles. Okay, that is the secretory tapetum. And next coming to the functions of tapetum. See here, this endothelium, the middle layers, tapetum. Okay, so this is the amoeboid tapetal cells and secretory tapetal cells. Function of tapetum. Tapetum plays an important role in the development of tapetum generates before microsporogenesis. Okay, so this is the tapetum plays an important role in the development of microspores. Mainly, tapetum degenerates before the microsporogenesis. Because of that, the pollen grains produced are sterile or abated. Tapetum also helps in the transport of food material to material to the inside of the anther. So it is helps in the transport of food material. That is the second one. And after that, they secrete the enzyme callose. Okay, the tapetum cells secrete the enzyme that is callose. We are called that is beta-1,3 gluconase, which dissolves the callosic wall of the tetrad and set them free. Okay, when after the development of pollen grains, they are formed as tetrad. So that is callosic wall. So dissolve that wall that helps in that callose enzyme helps in that dissolve. Okay, so that is the third function of tapetum. Tapetum is involved the synthesis and release of the materials which take in the deposition of trypsin and pollen kit. That is the fourth function. And also, tapetum initiates the process of abortion in pollen in cytoplasm, male sterile lines. Okay, this is the tapetum functions. We are coming to the here tapetum released amoeboid tapetum and function of tapetum. So mainly that main function is it provides nourishment, that is food material supply. Okay. So whereas that developing pollen grain gives the nourishment and it helps in the formation of exine also. In the pollen grain structure, outer is called exine. It helps in the formation of exine. It helps in the transport of food material and formation of pollen wall also. Okay. Next, sporogenous tissue. That microsporogenesis, primary sporogenous tissue gives rise to microspore mother cell. Okay, so that sporogenous tissue going to divide and gives the microspore mother cell. So the primary sporogenous cells may directly function as microspore mother cells. They undergo several mitotic divisions to form microspore mother cell. Each microspore MMC undergoes meiosis. MMC means microspore mother cell. Undergoes meiosis to form microspore tetrad, which separate out to form microspore or pollen grain. Okay, it that is mother cell. It is a diploid in stage, whereas that is going to meiosis. It forms the 4 haploid microspores. The process of formation of microsporogenous tissue is known as microsporogenesis. Okay, it's very important. What is microsporogenesis? From the microspore mother cell, that it going to meiotic division and forms the 4 haploid microspores. So that is the haploid microspores. So that's why that is called from the sporogenous tissue, the 4 haploid microspores are formed. This process is called microsporogenesis. All the sporogenous cells of an anther cells are capable of forming microspores, but usually some of these cells disorganize and provide nutrition to the active spore mother cells. Microsporogenesis. Next, coming to the sometimes that what is the cytokinesis? The process of formation of microspores from a pollen mother cell to meiosis is called microsporogenesis. Undergo meiosis and form the tetrad arranged in a cluster of 4 cells. Each cell of sporogenous potential to form a tetrad. So each cell is a microspore cell. See here, and we mentioned anti-hydration of the spores associate and deposit into pollen grains. After that, pollen grains are released. Okay, so these pollen grains released with the dehiscence of anther. So the development is successive type and simultaneous type. How that it will go that due to meiotic division, the micro. Please mute yourself. It's very important. Okay. During the meiotic division, the microspore mother cell going to it forms the it will wall formation may take place in 2 ways. That is successive type and simultaneous type. What is the successive type? And in this type, the wall formation a cell plate is laid between takes place of 2 daughter nuclei. Okay, in process of meiosis, that wall formation takes place. First, a cell plate is laid between the 2 daughter nuclei after the meiosis 1. So, in the meiosis 1 and meiosis 2 takes place between the after the meiosis 1. That between the 2 daughter nuclei, that cell plate is arranged in between the 2 cells. This wall gradually extends towards the periphery on both sides, dividing the cell into 2 equal halves. That is the successive type. Okay. Each cell of the dyad thus formed new now undergoes meiosis 2. So then 4 haploid spores are formed after the meiosis 2. That is the successive type. Okay. I think all of you understood clearly. This is the wall formation is 2 types, successive type and simultaneous type. Okay. All are you understood successive type? Okay. So then what is the simultaneous type? Successive type లో ఏంటంటే ఆ 2 డాటర్ న్యూక్లియై మధ్య సెల్ ప్లేట్ అనేది ఫామ్ అయిపోయి అది ఎక్స్టెండ్ అవుతుంది. సో ఇన్ బిట్వీన్ ద 2 cells అది ఎక్స్టెండ్ అయిపోయి ఏమవుతుంది అంటే it dividing the cells into 2 equal halves. So each cell of the dyad thus formed now undergoes meiosis 2. So then 2 to 4 haploid forms are developed. That is the successive type. In the simultaneous type, this type is common among dicotyledons. Here there is no wall formation after the meiosis 1. That is the difference between successive and simultaneous type. As such, instead of dyad, a binucleate cell is formed after the meiosis 1. Directly, no cell plate is formed in that simultaneous type. Directly, no wall formation after the meiosis 1. So, as such, instead of dyad, a binucleate cell is formed after the meiosis 1. So, that the 2 daughter nuclei undergo second meiotic division, and which is usually synchronous. So, the wall formation begins in the form of 4 peripheral constructions, which gradually extend from periphery towards the center. That is the simultaneous type. Okay. Yes.
Madam, shall we wind up?
I shared the photo.